Hello, everyone. Good afternoon, and welcome to our yet another offering of Trainee-to-Tenure Track Program at Gladstone Institutes. I'm Sudha Krishnamurthy. I'm the Director of the Postdoctoral and Graduate Student Education and Research Development Affairs at Gladstone Institutes. And this is our fourth offering of our series called Trainee-to-Tenure Track. The goal behind this program, as a number of you have attended before, is to help our early career scientists, primarily postdocs, senior postdocs and senior graduate students who are thinking about transitioning into academic careers to enable them with the skill sets that would make this transition easy. For today's workshop, we have Dr. Robert Mahley, who's our President Emeritus and founder of Gladstone Institutes, who has, in his exemplary career, has provided the art of lecturing for over 25 years. One of the things I'm very passionate about, Dr. Mahley, is how much he cares about each of his early career scientists, how much time he dedicates training the next generation. And you can see that passion and interest as he lectures this. And our next workshop, which is the following Monday, which is on Chalk Talk. So I hope many of you would join for that as well. Before we get started, and I hand this over to Dr. Mahley, just a few things to remember. We have a fairly big group. We are expecting over 100 people, and they are from different institutions, from the Gladstone Institutes, from UCSF, from UC Berkeley, from Lawrence Berkeley, from UC San Diego, and potentially from UC Davis as well. So we are recording the series. We encourage everyone to stay muted during the actual presentation so we can keep the background noise minimal. There will be breaks during the session, during the presentation by Dr. Mahley, and there will be questions, time for questions at that time. If there are any technical challenges, or if there's any question you would like to ask the speaker, feel free to direct message through the chat to Emma Doctors, my colleague, or myself, and we can review them and make sure we bring it up during the break periods for questions. With that, Dr. Mahley, it's all yours.
Okay. Well, thank you very much, Sudha. I really appreciate your leadership in this program and the way we continue to launch the careers of young scientists. And for all of you joining this particular session, I'm very pleased. It's the first time we've really linked up so many different institutions around actually the whole state of California. And so that's just great. So I'm going to activate the screen and start my lecture. So as Sudha said, I've been teaching the art of lecturing, I guess, something like 25 years. It's a topic of great interest to me. And so I've tried to learn from others what are the best practices, what works and what doesn't work so well. Now, we entitle this particular lecture series, The Art of Lecturing, because giving a lecture, an oral presentation, is very much like any other art form. To be good in music or dance or paintings or whatever art it might be, you can only get good at that skill by practicing. And so that's one of the messages that I would like for you to remember. Practice, and then practice, and then practice some more. Second critical thing is to have someone to critique your work. That's, of course, true for all arts, as you know, but practice in front of someone who will be honest and actually tell you the pluses and the minuses. You have in your handout, and I think you have all received this, one form that I like very much to use when I consider critiquing a talk, and I critique a lot of talks at the Gladstone, which is great fun. But you look at the content, you look at the organization, the lecturing style, how well prepared the lecturer is, and then, of course, the visuals, the graphics, which are very important. Now, you're going to develop your own style. There's really no right or wrong, but I do believe that there is good, better, and best practices. And of course, all of us want to be our best. So let's begin. And as I said, the purpose of this lectures is to consider best practices for effective communication of your research. And we're going to consider elements of a successful lecture, the goals of a scientific presentation, how to structure your story. Your lecture is a story. It has a beginning, a middle, and an end. We're going to discuss how to build that story. And then importantly, toward the end, we will talk about visuals or graphics that will make or break a talk. So what's the purpose of a scientific lecture? There are, of course, two obvious reasons that you know. That's to convey information and to convince others. Convince them by being an effective communicator. Now, we don't often think of scientists as having a product, but in fact, scientists have a product, and that product is information, it's our data, it's our conclusion, it's our insights. And I would like to suggest to you that 50% of your success is going to be dependent, of course, on the excellence of your data, but the other 50%, your ability to present your findings and your results. I would suggest to you to consider that you work as hard on your talks as you do in the laboratories, and that those that are most successful are those that acquire all of the skills, including the art of lecturing. So let's consider the general elements of a successful lecture. Now, all of us present to very intelligent audiences. And in fact, we must consider that it's our fault if they don't understand our lecture. Everyone on this Zoom call I know is capable of understanding any lecture on any topic if, in fact, it is delivered well. And that's our responsibility as the lecturer. A second point for your consideration is don't make your lecture a mystery story. Make it easy to follow. And I'm going to suggest right up front that you remember the next three phrases. If you remember nothing else from this lecture, remember these next three phrases that guide us in giving a good talk. The first of all is tell them what you're going to tell them. That's the introduction. That's the beginning of your talk. It's leading to the message, which we'll define in just a minute. Second, then you tell them. That's the facts, that's the data. That is supporting your message. And finally, third, tell them what you have told them. That is the conclusions. Now, I'll say a lot more about that in just a minute. Third, remember the KISS principle, and that stands for keep it simple. Listening to lectures, as I say here, is hard work. So help your audience to understand. Don't load your talk up with a lot of extraneous facts. Just tell me the essential points, the essential facts that I need to understand in order to understand your research and your results. Not everything that you know on a particular topic. You're the expert on that, but I don't need to know everything. I just need to know what I need to know to understand. And fourth, you are creating a story. It has to be a story that's easy to follow. Manuscripts are where you document details, but in a talk, you tell the audience only what they need to know. Never, in fact, use a figure or a table from a manuscript. They're usually just too detailed. They're loaded up with too many facts, footnotes, and so forth. For example, in a talk, you almost never go into the level of depth of detail, like, oh, I used 1 molar sodium chloride at pH 7.2 for 28 minutes. Usually in a talk, those are things that I really don't need to know. Now, let me share with you just very briefly how I go about preparing a lecture. The first thing that I do is to try to determine the message. And the message is what I want you as the audience to learn and to remember. I use this message to build my story. And then I collect all of the data, all of the facts that I need. What in the background do you need to know to understand the research that I'm going to present? What data is required? And so the way I collect that is just a series of slides, and it may not be real slides. It may simply be sheets of paper on which I've made a note of something, a drawing, a set of data that I want to particularly summarize. I end up with this stack of pages, figures, formal or informal. Then I can shuffle them around. I can organize the talk, but I'm organizing the talk based upon the message that I want you to hear about, that I want you to learn about. That serves as an outline, actually, and helps me to develop the visual aids that I need to present for the talk. And fourth, I write out every talk that I've ever given. Now that's I know most people don't do that, and this is, of course, a personal choice. If you're giving a 10 or a 12-minute talk, like an abstract at a meeting, you must write it out because every word counts if you're going to get your message across. So write it out in considerable detail. Now, you're not going to read it. That's not the purpose of this. The purpose of this is to get you to organize your thoughts succinctly and clearly, that you can present rapidly. Now, why do I write out my talks? Well, I get very nervous when I present. I get anxious. I forget things that I really want to tell you. And so it helps me to have a text. It's really my safety net. I don't read it. Sometimes I don't even refer to it other than to glance at it, but it's there. If I feel that I am not giving the message the way I want to give the message, then I can see my notes. So let me repeat. The goal of a scientific presentation, twofold: send a clear message and to tell a clear story. What do I mean by the message? That is, as I've already said, what I want you, the audience, to learn. And second, what I want you to remember. Telling the clear story, it is a story. It must flow logically. The story explains the message. The story validates the message. The story makes the message clear. Now, one of the biggest compliments that any lecturer can receive is to hear someone going out of the lecture and hear them to say, I learned X, Y, and Z. That was the message. That's what they learned. And usually, if they can say that, they will also say, I'm glad I attended that lecture. Well, let me say more about the message and what I really mean by the message. It is the main point of the talk. The message should be stated on a slide early in the talk, It's usually fourth, fifth, or sixth slide in most talks, depends on the length, of course, but it's early in the presentation. It should be able to be presented as a sentence, that is, with a subject and a verb, although you don't put it as a whole sentence on a slide. And it will be repeated, it should be repeated 2, 3, 4, 5 times. In the part of the lecture that I'm going to give you just little snapshots of, I will use my message slide 5 times. And that will serve as my outline, it will serve as my guide, and it will allow you, if I'm effective in my talk, for you to know exactly what I want you to learn. As I said, don't make your story a mystery story. Tell me right up front what you want to tell you, what I want to tell you. That is, I'm giving you the conclusion up front. That's the message. Most people don't do that. And in fact, many people say to me that, well, if I do that, if I give the message up front, if they know what I'm going to want them to learn, they'll be bored. They won't listen. Well, in my opinion, that's absolutely not correct. I promise you, try it. It's just the opposite. Because if you tell me what the message is, I'm going to know what to focus on. I'm going to be able to follow more easily. I'm going to be able to pay attention, real attention to the data. I can relax. I can think about what you're really saying because I know where you're headed. Now let's talk about putting the story together. That is the structure of the story. As a story has 3 parts. It has the beginning, it has a middle, and it has an end. This actually is a famous formula. Sounds very simple, but it's a famous formula that almost all CEOs and executives of Fortune 500 companies take such a course. One such course is known as the Toastmasters Club. I participated many, many years ago, and this is exactly what they teach you. 3 parts. The beginning, that is leading up to the message. Tell them what you're going to tell them. The middle, tell them, that is, prove the message with facts and with data. And then the end, the conclusion, tell them what you told them. Repeat the message and use the same words. Now, this morning, actually, I heard comments from Eli Lilly, which they had just had a successful clinical trial. And you could see that this was exactly what this spokesperson had been taught, a beginning, a middle, and an end, because he said, Trulicity lowers blood glucose and decreases body weight in type 2 diabetes. And then the middle, he gave the facts. In the latest phase 3 trial demonstrated X, Y, and Z, and so forth. And then he ended that message with exactly the same words, the message that he began with, Trulicity lowers blood glucose, decreases the body weight in type 2 diabetes. So let's talk more about the beginning. Remember, the beginning is a step-by-step build-up to the message. But before I give the message, there are several things I need to do in the beginning. I need a throwaway comment or few comments. I'll talk more about that in just a minute. And then I need to introduce to you the general topic. That's the title of the lecture. That's what the talk is all about. And then I have to provide you with enough background, enough facts, definitions in some cases, maybe a statement of why the topic is important so that you can understand the message. Now, you usually can't get the message in just the first couple of slides. The audience typically doesn't know enough of the facts about where you're headed. So the background is really critical if they're going to understand the message. So let's back up a minute. Let me tell you about throwaway comments, the first 15 to 20 seconds of a talk. Good afternoon. My name is Bob Mahley, and I'm a senior investigator at the Gladstone Institutes. And it's always a pleasure to speak to the neurology department at UCLA. Well, why are these two sentences that I just gave you, why are they important? Well, for one thing, it allows the audience to settle down. You've just come in from the lab, I'm sure. Maybe the experiment wasn't going so well. You need time to settle, and you need to tune into me. You need to understand my voice, my style, and so forth. So this gives you just a little opportunity. And also, it acknowledges the audience. I'm glad that I'm here. And of course, you mean it. So now let's go a little further into the introduction. And that's the general topic. That is the title of the lecture. I like it simple and direct. Tell me what the talk is about. I don't like funny. I don't like cute. I don't like references to Star Wars. A lot of times I don't get them. Just tell me what you're going to talk about so that I can understand it. Now, in order to illustrate some of the elements of a lecture, I'll give you parts of a lecture that I recently gave. And so I'll give you just a little snippet of the beginning and then a little later, the middle and the end. So the first slide in my talk is the title slide. Apolipoprotein E4 and Alzheimer's disease, that's the general topic. Mitochondrial is associated with mitochondrial dysfunction and altered CNS metabolism. The title is informative. I'm going to tell you about a protein. It's a protein called apoe. Specifically, it's called apoe4. Now, you may have never heard of this protein. Doesn't make any difference. That's okay. Because if I do a good job, you're going to know what you need to know. And then I'm going to tell you how it fits with Alzheimer's disease. Alzheimer's disease is a terrible disease. It's increasing in prevalence. And so that makes this topic to most people. Many of you have grandparents and so forth that may be having some memory problems related to Alzheimer's. And so I'll tell you just enough facts so that you're reminded about what Alzheimer's is. And then I'll tell you how this protein, apoe4, impacts mitochondria and CNS metabolism. So you know the general topic from the title. And now I must provide you with the background leading to the message of the talk. The background is what the audience needs to know to understand my story. In that background, I've got to define all the terms that I'm going to use. I've got to set the stage for the research that I want to present to you. And remember, in this process, I am leading you up to the message. Now, remember, as you put together the story, what's common knowledge to you may not be common knowledge to everybody in the audience. We speak to a lot more general audiences than we tend to believe that we speak to. And a lot of times people really don't understand. Don't use jargon, don't use laboratory shortcuts. And don't, what I refer to, don't use alphabet soup unless you define that alphabet soup very carefully. Now, what do I mean by that? A few years ago, it seemed like I attended a whole series of lectures where the important data that was being presented, they referred to as ChIP-seq. Well, at that time, I'd never heard of ChIP-seq. And so I missed the point of more than one lecture until I learned. But that speaker very quickly and very easily could have told me what ChIP-seq was and any of the other alphabet soup terminology jargons that we use. Put it on a slide and define it. That's the best way for people to remember. For my talk, the audience that I've just told you about, the audience needs to know what apoe is. You need to know that there are three forms, three isoforms. ApoE4, it was in my title. ApoE4 is most important for Alzheimer's disease, and that its structure determines its function. You got to know that before I start talking about apoe. You need to be reminded about something about Alzheimer's disease, a neurodegenerative disorder associated with dementia. And importantly, this puts apoe in context, 65 to 80% of all patients with Alzheimer's disease have at least one copy of the apoE4 allele. And this work does have therapeutic potential. So let me show you what I put into the background to bring you up to speed. So I'm going to show you now, first slide, title slide. Second slide, I have to define apoe. There are three isoforms of this protein in humans, apoE2, apoE3, which is the most common form, and apoE4, which is not truly rare. The forms differ by a single amino acid interchange that occurs at residue 112 or 158. Looking at the difference between apoE3, the common isoform, and apoE4, it differs by this single change of an cysteine to an arginine, and thus apoE4, as I will show you, becomes a major risk factor for Alzheimer's disease. That's the second slide. The third slide is Alzheimer's disease, most common devastating neurological disease facing humankind worldwide. It's a dementia, robs the patient of memories, results in a loss of CNS neurons. Prevalence is increasing. There is no effective treatment as of now. There are 5 drugs on the market, none of which are truly helpful. The next slide. Alzheimer's disease is a complex disease. There are multiple causes, multiple pathways that are involved. For example, one pathway involves amyloid plaques, a buildup of amyloid beta peptide that accumulates in the parenchyma of the brain. Second, neurofibrillary tangles made up of the protein known as tau. This forms tangles within the nerve cells, preventing normal function. But today, I'm going to focus my comments on apoE4, which has a direct toxic effect in causing neuronal cell death. Thus, the focus is E4 and Alzheimer's. Now, depending on the audience, if it were the neurology department at UCLA, UC San Diego, wherever it might be, I wouldn't necessarily show this type of a slide. But if it's the chemistry department at Rutgers, they would want to know more about what the brain actually looks like in Alzheimer's disease. So now this is the 4th or 5th slide in the presentation. It's still background, shriveled brain, loss of neuron, white matter and gray matter in the brain. And so now, with this background, 4 or 5 slides, you're ready to hear the message. I am going to tell them what you're going to tell them. This is what I have been leading up to. Let me just remind you, the message is stated up front. It's going to be on the 4th or 5th or 6th slide in my talk. It's going to be on a slide. I'm going to use that slide more than once because this really is my message, really is what I want you to remember and to learn. And it's going to allow you to relax, hopefully enjoy my talk, and just focus on the quality of the data. Is it good? What are the holes? Where's the problem? So what is the message of the talk that I have just set up for you? The message, now this is not the slide I would use, but this is putting into words what the message of my talk is. ApoE4 assumes an abnormal structure, point 1, that causes it to undergo neuron-specific proteolysis, number 2. 3, generating neurotoxic fragments that cause the mitochondrial dysfunction, cytoskeletal alterations, and 4th and 5th, leading to neuropathology. This is what I want the audience to learn, what I want them to remember. Now, it's my job when we get to the middle of the talk to prove to you these points. It's your job to see if I do and see how interesting the data is or not. But I'm going to convert this message. This is not a good slide. You'd never use a slide like this. But this is what was in my mind that I wanted to tell this audience. But I converted into a usable slide that actually becomes the outline for my talk. I build the talk around this slide now. It's exactly the same thing that I just said. ApoE4 forms an abnormal structure that is associated with neuron-specific proteolysis, the generation of neurotoxic fragments, leading to cytoskeletal alterations and mitochondrial dysfunction, finally, neuropathology. So this is my message slide. It's the 5th or 6th slide in this talk, depending upon how much detail I give in the background. And I'm going to use that slide over and over again. For example, when I've gotten down through the first 3 points, and now I want to focus your attention on mitochondrial dysfunction, impaired oxidative phosphorylation associated with ApoE4, I highlight that part of the outline. I'll do that step by step as I build the story. Now, we're at the major part of the talk, and this is where you wanted to get right away anyway, because this is your data. These are the facts, the things that you really wanted to share with the audience. That's the middle, and that is the tell them part. I've spent a lot of time talking to you about the beginning, because in my opinion, that's where most talks fail. Not giving enough background so that I actually can understand where you're headed, what I need to know when you start telling me about the facts and about the data, so that I have to have enough background so that I can understand your data. Now, in a 30-minute talk, what proportion of the time is allocated to the beginning, middle, or end? Well, it of course differs a little bit depending on the topic, but the beginning 30-minute talk may take 5-7 minutes. The middle, where you want to get the data, 15 minutes, maybe 18 minutes, and the end, 5 to 7 minutes. That's just a typical way that I would suggest you consider. So what I'm showing you is that the beginning consumes about 20 to 25% of your time, because I've got to know enough to understand your data. Now, how many slides will I use in a 30-minute talk? Well, ideally it's 30. That's a minute a slide. Takes about that long to describe most data, but it's probably going to be like 35 slides. Some of them are quicker than 1 minute. So probably 35 slides for a 30-minute talk. That's probably about right. If you have 45 slides, you're going to be in trouble of getting your point across, in my opinion. So let's remember, we're telling a story. We're at the fun part now, the middle, where you describe the experiments, but you've also got to link the experiments. It's a story. It's got chapters and verses that you have to go through for it to logically follow. Now, each experimental set of data that you're presenting in the middle of the talk has to be introduced. And so we usually follow something like the scientific method. That is, we present a question, as I will show you now, something about a question. It could be a hypothesis. And then you describe the experiment. That may include methods and the results. And finally, an answer. This is what that experiment has shown. But I would like to suggest a little different twist to that. I would suggest that after you state the question, you give the answer, what you are going to show by your experiment. And so here again we are. Tell them what you're going to tell them, then tell them with your scientific results, and then repeat the answer. Tell them what you told them you were going to tell them. For example, how does the single amino acid interchange in E4 that I mentioned to you contribute to neurotoxicity? That's the question. The answer is, I'm going to show that this change in this single amino acid causes E4 to undergo proteolytic cleavage generating fragments. Then I present the experiment and I conclude what I just told you I was going to tell you. The second part of the tell them is to link the experiments to each other and to the message. It's a story. It must flow, as I've said several times. Now, there are various ways to link experiments to each other. Sometimes they're spoken links. So I've shown you data at the level of the tissue. Now let me go to the molecular. Or I have shown you data from a series of in vitro studies. Now let's go to in vivo studies where I've used the mouse model for whatever. Or, so to go back a little further, let me do thus and so. Or simply now, the next step in understanding this problem was to, and then you've introduced the next experiment. But what I actually like to do is to repeat the outline slide, which you will recall is the message slide, to guide where you are going, but to also remind you of where you've been. So here's the type of slide. You've seen it now several times. I've talked about the abnormal structure. Now, when I completed that, there'll be a series of experiments that I'll relate to you. It undergoes, because of this abnormal structure, neuron-specific proteolysis. And then I show you the data related to the protease that might be involved. And then I go to a next experiment, the generation of the neurotoxic fragments that are responsible for mitochondrial dysfunction and so forth. So I use this message slide. I will use it 5 times. I used it 5 times in the talk that I gave. So in summary then of the middle, the tell 'em part, I would suggest that you try to use this approach where you state the question that your results are going to investigate, and you give the audience the answer. You tell them what you're going to tell them, and then show them the results, and then tell them what you told them you were going to tell them. Linking all this together and back to the message of the talk. Now, how do I prepare the middle of the talk? But it's exactly like I said in the overall putting together a talk. I use the message slide as a guide to develop the story, and then I collect the data. Here again, I lay out all the data that I have to tell, that I want to tell. Most of the time I can't. There's just not time to go into all of it, but I have it all there. I have it on sheets of paper. Maybe they're rough figures. Maybe it's just a few words on a piece of paper, which will become a slide. And I start shuffling those, actually designing the slides to develop a logical flow. So let me go back now to my demonstration lecture, a little part of it, to illustrate using the message slide to introduce a new topic. And this is actually the first set of data that I'll present in that talk. So this is the slide. After I give the message slide, I've just shown this, but now I highlight this because this is the first set of data I want to share with you, and that is about the abnormal structure of apoe4. And so I say something like, let me begin by comparing the structure of apoe3 and apoe4 and describe for you the abnormal structure of apoe4 that is associated with the abnormal effects on nerve cells. And so then the next slide that I show is this slide, and I say something like, and as I have said, apoe3 and apoe4 differ by a single amino acid residue occurring at residue 112. ApoE3 has cysteine at this site, whereas ApoE4 has arginine. Now, during the actual lecture, I would present the data then that defines this structure, this profound, actually it's a profound change in the structure and the function. I would show how the various residues are involved in the amino terminal and the carboxy terminal interacting in ApoE4 and how this is associated with neuropathology. So that's the next slide. And then I talk about the relevance of this data to a potential therapy, and that's using small molecules called structure correctors, which can insert themselves, break this interaction, converting ApoE4 structure, as it says here, to an ApoE3-like structure and reverse the detrimental effects of ApoE4. And at this point, then, I would show data defining how these small molecules reverse the detrimental effects. And then I would repeat the message slide wherever I am in the topic, highlighting that particular area because of the abnormal structure, neurotoxic fragments having an effect, and so forth. So to this point in the talk, I've actually used this slide 4 times. Abnormal structure, proteolysis, fragments. I focused your attention on mitochondrial dysfunction. And now we're to the end of the story. Tell them what you told them. Repeat the message of the talk. use the same words. Don't make it hard on me. Make it easy. The day's been long. I'm a little tired. I really want to understand what you're telling me, but help me. And then you can go on and tell me very exciting things about the implications and future directions. And finally, I will end with this slide in which I have told you how the structure of apoe4 is abnormal, causing it to undergo neuron-specific proteolysis with the generation of neurotoxic fragments. Mitochondrial dysfunction is one of the outcomes, and then we have neuropathology. So what else can go at the end of your talk? And it depends upon the type of the talk. If it's a job interview talk, it's a little different. If it's to a group of specialists, it's different, or to generalists. But you can have conclusions. What's the interpretation of the data? If something's a little controversial, what does it mean? How does it contribute to biomedical science? Have you proven the hypothesis? Have you answered the question? Now, sometimes you won't. Nothing wrong with that. For example, I don't know what the protease is that generates these neurotoxic fragments. Dr. Yadong Wang at the Gladstone is working on that. And we don't know what that protease is right now. So we know there's a protease. We know it generates these fragments, but we don't know what it is. That's fine. I need to make sure that you understand that. And then if it's a job talk, you need to talk more about future studies. So in summary then, we've considered the structure of a research talk. We want to tell a clear story, and we want to make it easy for the listener. In the beginning, we've built up slowly. We've built with throwaway comments and with a lot of background to develop the message. Slide 4, 5, 6 in the talk. And then you're to the middle. Slides 7, 8, 9, 10, 11, 12, 13, 14, 15, whatever, to describe the experiments, to link them. You end by repeating the message and whatever else you want to conclude. So why bother honing the skills? Isn't the most important thing my science and my data? Yes, that's true. But it's also equally true, you got to be able to present it. And if you can't, why did you do it? So work hard on doing it. So let's consider the importance of preparing a good oral presentation. First of all, consider every presentation is a job interview. You never know who's in the audience. As I've said before, we don't usually consider that we as scientists have a product, but we have a product. That's what we're doing. We're generating data, insights, conclusions, and now we got to sell our product. We want to sell it in a convincing, honest, direct way. I'll give you one personal example. It was a couple of years ago. I heard an excellent talk at a Gladstone RIPS seminar. I had firsthand knowledge. It wasn't in my area, but it was a really good talk, and I enjoyed it very much. Well, it turns out this graduate student had applied for a job at another university for a postdoc position with a distinguished professor. She hadn't received a response from this professor, but I knew what she was doing and I liked it, and because she had presented it clearly. And so I called the professor and I said, you know, you ought to move that application a little higher in your stack and take a look at Dr. So-and-so. And she's now completing her postdoc with this distinguished scientist. She did it on her own. Don't think that I'm taking credit for her success, because I'm not. But I did help to get her application to the top because I understood her research. Every presentation, second, can introduce you to a new mentor, an advisor, an employer, a collaborator. I was fortunate when I was an MD-PH student at Vanderbilt to give an oral abstract presentation at the American Heart Association meeting. And that led to me meeting a distinguished scientist who was in the audience, who became a mentor, an advisor, contributed to my employment, and became a collaborator. I introduced myself to him after my abstract presentation. I needed to learn a particular set of skills related to protein chemistry. And because he had heard my presentation, understood what I was doing, he invited me to the NIH to spend a couple of weeks learning the technique. Ultimately, when I completed my MD PhD, he facilitated the recruitment of me to the NIH, where I started my independent career. You never know what this presentation is going to lead to. Third, every presentation focuses your attention on the next set of studies. So as you put it together, you will realize sometimes for the first time what's missing to complete the story. Or what did the audience ask you, maybe you hadn't thought about, that you needed to provide. Now, before COVID, we had a guest speaker at Gladstone. She was a professor from UC San Diego. She was asked a question after the lecture, and she stated, you know, that question suggests a new experiment to me that I hadn't thought about. And then she went on to say, that's why I like to give lectures. So that's the type of experience you can have. And then next, every presentation can serve as an outline for the manuscript, because you're compiling tables, figures, illustrations. This is not wasted time to put together a detailed, good presentation. It becomes an outline for a manuscript many times. Every presentation can lead to the next presentation. Why? Because program organizers want to program excellence. And so what are the two critical questions that scientists ask as they sit around putting together a symposium or a keystone or whatever it might be? Well, first of all, they say, well, is his or her science solid? The answer has to be yes, or you won't get invited. But the second, equally important question is, does the person give a good talk? I promise you, this is what's said when you put together a program. And if the answer is, oh yeah, the data is good, but it wasn't so clear the last time I heard her speak, well, he or she is not going to get invited. So establish yourself for excellence, for your science, of course, and for your ability to present. And it's not easy. It's hard. It's hard work, but it's worth it. There's lots of reasons to hone your skills. And so now I'm going to stop, and I will open it up to questions. Please feel free to unmute yourself and ask me directly whatever you would like to ask. And we'll have several minutes if there are questions for you to ask them, and then we will continue and go into visuals which will make or break a presentation. Equally as important as anything I've said thus far. So please Any questions? I guess I can see hands, or you can just speak up. I'm not entirely sure. Unmute yourself and ask the question, or maybe there's some in the chat.
There are no questions in the chat as of now, Bob.
Okay. Questions from the audience. What about answering questions? That's a part. I haven't put anything into this lecture about that. Probably that's a mistake on my part. That's the toughest part, isn't it? How do you handle questions? Do you ever say you don't know the answer? Somebody talk to me.
I feel bad that I'm the only one responding. Come on. Enough of us here.
Yeah, if you don't know the answer, that's what you say. You know, that's a really good question. But you know, I'm going to have to think about it because I'm not sure I know how to answer it. Could I talk to you after the lecture?
So we have a few comments. Oh, this one question.
Okay.
Stephanie, do you want to unmute yourself and ask the question? I want to encourage people participating.
My question is, if we have an opportunity to give, here, I think I can video as well. Yes, hello.
Yes.
When we have an opportunity to give a rather brief talk, say 15 minutes or even less, what would you recommend of the principles that you've shared so far around what to retain, especially if we already have been accustomed to giving longer talks?
Yeah, well, 10 or 15-minute talks, of course, are very common. And you have to then determine one message, one major message. It's pretty hard to, and you couldn't give the talk that I just demonstrated for you. There's no way. The only thing I could have possibly done would be to talk about mitochondrial dysfunction. And so I would have to very quickly say, the structure of apoe is abnormal. It generates neurotoxic fragments. Here's the gel that shows them. That's all the time I got. That's a 1-minute part of the 10. And then I go into the proteomics of mitochondrial dysfunction and talk about the excitement of the subunits that are decreased in the electron transport chain and one seahorse experiment. That's all you got. But you gotta the beginnings of a good story, and it'll open the door for further discussion, either at the time of the presentation or afterwards. Does that help, Stephanie?
It does. Thank you.
Okay.
Hello, Bob. Thanks for the talk so far.
Sure.
Hopefully I'm audible through this message.
You are?
Okay, perfect. So one question that I had is when you're giving a talk at the end and during the Q&A, there's, I guess, sometimes I struggle with balancing specificity versus simplicity in terms of how detailed I get in answering a question. How might one balance that in like a seminar, for instance?
Yeah, that's the questions. As I said, I should have talked more about questions, asking and answering, and actually I do have something in one of the handouts, which I do. It's in the handout. It's under the 12 deadly sins, the don'ts of lecturing. Actually, there are quite a few comments there. Don't fail to repeat a question if likely someone in the audience hasn't heard the question. That's not your question. I'll get to your answer. Don't hesitate to restate a question if the questioner is unclear, or if you are unclear as to what the questioner really is trying to get at. As I said earlier, don't fail to admit that you don't know the answer to a question. And don't fail to know your audience with respect to knowledge, scope, and breadth. So those are some things. Now, yours question was a little more specific, and that is about how specific to get. For one thing, you got to judge your time. And most of the time, especially when questions are asked during the middle of a seminar, which they often are, actually some of those are the best discussion points, but nevertheless, you want to get to the end of your data. But you can't ignore the question. You've got to respond, and you'll have to judge how long it's going to take to answer the question. But don't be fearful to simplify the question. They can always ask again, or if they didn't get enough, they'll come up to you after the lecture. But you have to judge, you have to pace yourself, and you have to be, actually, you have to be willing to cut them off at times. If you have a person in the audience that's really bugging you, that asking a million questions which you don't actually think are on target, then you have to be polite. There's no question about that. It's not anything that I'm even suggesting, but you have to oftentimes say, that question is going to take a little longer to answer. Would you mind if I talk to you after the talk about that? Because that's really an important question, but I think I need to give you some more details than maybe I have time right now. So I'm not, Yoshi, I'm not sure I've answered your question, but at least I got an opportunity to talk about questions. I turned your question around to suit me. So you can do that, and I don't think you were offended that I did that. But nevertheless, I got back to your question, or tried to.
Well, I think that that in itself was very helpful for me, Bob. Thank you.
Okay, yes. Other questions before we continue?
I have another question.
Yes,
about excellent talk, by the way. No, it's probably better than any talk I'll ever give. Do you have any advice? I find that I slip into this problem a lot. Everybody wants to feel as though they've been really productive and show off how productive they've been. So they put too many slides in their talk that they'll probably never have time to get to. Do you have any just general advice on that front? Should you always underload your talk, or how do you convey that you've done a lot without wasting a lot of time, I guess?
Yeah, this is a critically important question, Jason. The number of slides are killers in talks. So you figure out the number of minutes that you've been allocated, and you add a few more slides than the number of minutes. Just like I said, a 30-minute talk, 35 slides, that's probably about the limit. An hour talk. I never planned for an hour talk. I always planned for a 45 minute talk, which means I'll have 50 slides, something like that. And it almost always works out that way. You've got to limit the number of slides. Now, I do have a pet peeve that will contribute to the answer, and that is when I hear presenters stand up and say what all they're going to talk about, and they waste a lot of time listing all this stuff, and they say, oh, and if I get time, I will get to this particular interesting part of my work. Well, that irritates me to no end, because that means they didn't practice. They don't know how long their talk is. I know how long my talk is. This first talk I gave you was an hour long. It was an hour by my watch. It was an hour yesterday when I practiced all day. It was an hour. I didn't have to tell you after I gave a brief introduction, well, maybe I can get to talking about visuals. No, no, I know how long the talk is. So plan it that way. Did I answer your question?
Oh yeah, no, that helps
Okay.
a lot. I think I tend to be very verbose, so I should probably do under by 5 slides, not 5 slides more.
Well, Jason, if you're verbose, I know you, Jason, and you're great, but I would recommend you write out your talk, and then you can strike out every other phrase that makes it too long and you don't get to the meat. Okay?
All right, thank you.
Thanks, Jason.
Super helpful, thank you.
All right. Maybe we should go on. Well, good. As I have already said a couple of times, the visuals, the graphics, the slides will make or break a talk. I really believe that the quality of your slides really does reflect the quality of your work. As I've written here, poorly prepared, you haven't spent time, you put a bunch of stuff on the slides that you don't talk about, they're difficult to read. They really do detract from the excellence and actually your excellence. So consider the preparation and use of the slide. I consider it one of the most important parts of our discussion now today. I want us to think together about the best way to graphically communicate your story. Slides don't replace your words, but they're meant to strengthen the story. So let's look at some guidelines for visuals that I think might be helpful. First of all, do keep them simple. I think I got my arrow working now, excuse me. Keep them simple. Remember the KISS principle for the words as well as the graphics. Models are extremely helpful. It's cliche to say a picture's worth 1,000 words, but it is. Visuals are just a big help. Large enough to read. So now once we get off of Zoom, which is coming quickly, I hope, we're going to be back to auditoriums and conference rooms, and therefore you're going to have to remember what I'm talking about. On Zoom, you can get away with lots of stuff, but once you're in a conference room or once you're in a big auditorium, you got to worry about the size of the room, the size of the audience, and you've got to be able to use a laser pointer. I'm not so good with this pointer that's on my mouse. Laser pointer, I'm better, but you have to practice with the pointer. You have to lead the audience through the slide, and for heaven's sakes, don't circle with the pointer. This often makes people a little nauseous. So try to use the pointer in an appropriate way. Now, when you've got a laser, you know, people say that I circle with the pointer because my hand's shaking. Well, there are ways to get around that. For example, you can press your arm against your side. I don't really know whether you can see me or not, but and that steadies the pointer. Or you can actually rest your hand on the lectern, which you're usually standing behind, and use it. You can do it, but try not to circle. It really is annoying. Don't speak too long with the wrong slide, the wrong visual on the screen, because your mind wanders. It wanders anyway in everybody during a lecture. But if you got the wrong visual up there, keep me focused. Don't let me wander. Keep my pointer telling you exactly where I want you to be focused. Use transition slides for breaks. Don't just let things slide into each other. We'll talk about this in a minute. Typically, a good data slide should be on the screen for about 60 seconds. Now, there are some occasions where, transition slides, it's 5, 6, 10 seconds up there. But one of the more irritating things, another irritating thing, is if the slides go too fast because you realize you got too many. So if I just show them faster, it's better. Well, it isn't better. I always take notes during lectures. That means my head's down some of the time. And once in a while, a lecturer, I already missed one slide because I was writing from the last slide and I know he showed another one or she, and now it's gone. So I missed the whole thing. Sloppy slides suggests sloppy lab procedures. All right, lecture room. Be comfortable with your surroundings when we get back to our lecture rooms, our conference rooms. If you're in your home institution, of course, you can go in, you can feel comfortable in the room. When I'm visiting professor, I'm often shown around the university by a postdoc or a graduate student. And so during that, when I first meet my host, I say, sometime during the day, before I give my lecture, if we could just stop by the lecture room, it'd help me. And then I can see where the lectern is. If I got to worry about lights, I can see about the lights. I can see about how to connect my computer, so forth. Because you often come in at the very last moment, you're about to get introduced to give your talk and you're trying to get everything settled. If you're nervous like I am, you're even more nervous at that point. Second, practice. Practice with equipment if you can, with a knowledgeable colleague. I've already said this, but find one or more people that will be honest with you, that will tell you. Now, when I critique a talk, the first thing I say to the person that asked me to hear a lecture and critique it is, will you give me permission to be honest? I'm going to tell you the strengths. I'm not going to be hurtful, but I am going to tell you the truth if I don't understand something or if I think you did a lousy job. Will you give me permission? And if they won't, well, they always do, but if they wouldn't give me permission, then I wouldn't feel comfortable. Practice with a clock. As I said, my first lecture was an hour long. It was an hour long, period, because I knew how long it was. Now, how long does it take to prepare a talk? Well, 10 hours to prepare a 20-minute talk. That's generous. It's probably true. 24 hours. That's not sitting at one stretch, but putting everything together. It takes time. So another thing that irritates me is to hear, and it's often a full professor that says, oh, I got a lecture to give here in 15 minutes. I better go get some slides. Let me tell you, it's probably going to be a lousy lecture. If in fact, well, most of the time they're not actually being straightforward with that comment, but because they probably have thought, at least I hope so. Oh, one other thing about the lecture room and equipment that I forgot to put on here, and that is take your own pointer, your own laser pointer. Never trust the organization to have one with batteries. Always bring your own. Okay, general points about design of slides. Simple text, bullet points like illustrated here, not sentences. Make sure the design is readable. That's the right font size. You almost can't make the font too big. Use a slide from top to bottom, left to right, just like we read. Do not use tables from a manuscript. The title can describe the slide or it can draw conclusions. Or let's talk about the titles. Let me say more and give you some examples about titles. Every slide should have a title. Every slide. Okay. Two different types of titles generally. One is a description of the data. It may be method, actually, a method being presented on the slide, or it can be an interpretive statement of what the results show on the slide. I actually like this second point because it helps me to make sure that I understand what the presenter thought, at least thought, the data was showing. So let me give you a couple of examples, and they're examples from things that I have available to me. So of course it usually has something to do with apoe, but let me give you a couple of examples. For example, here was a slide that a presenter used in one of the Art of Lecturing courses where we actually have presentations and critique them. So don't worry about what it shows. These are actually those neurotoxic fragments that I was talking about, in case you really wanted to see them. And this is an anti-apoe immunoblot of the supernatant fraction from a human brain. Well, this worked. That was okay. That gave a lot of information. Some people don't know what these blots really are, so you need to help. But the title that I liked the best was that this is what the slide showed, that carboxy-terminal truncated forms of apoe are generated in the human AD brain. As I said, don't worry about what you're actually seeing here. I'm just trying to give you examples of types of titles. Now, the reason I like this type of a title is because some data that's presented, initially that ChIP-seq data, which totally threw me until I figured it out, I needed to know what the speaker thought he was showing. And so I have a difficult time with electrophysiology. Sorry, Jason. Jason's electrophysiologist, but I have difficulty. I really can't interpret all of those wiggles. And so I rely upon the title to tell me what the electrophysiologist believes the data showed. Okay, here's another example of a descriptive title. You choose the title based upon the audience too, because this particular slide, again, it's apoe, I'm sorry for that, and it's a water maze where it's a learning and memory type of test. The E4 mice highlighted here in blue, they really don't learn well over many trials to find where the hidden platform is, but the E3 mice do. And the title here was Performance of 6-month-old female E3 and E4 mice in the water maze. This meeting actually was one related to age and gender. So it made good sense to talk about age and gender in the title. But another title which is just as good and which I tend to use mostly is that apoe female mice perform poorly in the water maze. That's what it shows. E4 inhibits learning and memory. It's not right or wrong, the type of title you use, but you try to, excuse me, help the audience to understand your talk and to understand the data. Now let's talk about different types of slides. I've already mentioned model slides, a working model. If you can summarize your results in a model, in a drawing, a schematic, that's always best, because everybody usually can understand that type of a summary more easily than a list of words or phrases. In fact, I always ask my students and postdocs to draw the scheme that they're studying. What is it? What points are you trying to impact? Draw it for me, then I can follow it better. So let's talk about model slides. This is a version of the model that I showed you in my demonstration lecture. It's more complicated. This is for more of a specialty audience. where they really wanted to understand the amino acid residues that resulted in this domain interaction in apoe for the abnormal structure by comparison to E3. So this is a slide for more of an expert audience. Now, I can use a slide like this, same information, but simpler. You don't have all of those alpha helicals and beta sheet structures that can confuse people. Now you've got an amino terminal domain, a carboxy terminal domain, the residues that interact, the difference between E3 and E4, and in this case, how a structure corrector works. Much simpler. So you think about your audience as to what the level of detail that you really want to give. Now here was this was used in the Art of Lecturing course in which students come together and do presentations, and they're critiqued, and then they repeat their presentations to make them better and so forth. That's a much longer course than we're talking about today. But this was a beautiful method slide, absolutely beautiful method slide. He was studying HIV infection in rats and the methodology. He wanted to get through the methods quickly so he could really talk. It was a 20-minute talk. He could really talk about the data and the facts. And so this is the slide he used. And so he injected the HIV intravenously or interperitoneally and followed its distribution in the spleen, the brain, the thymus, and the brain in anesthetized rats. How more beautiful can you get? And what did that take? 30 seconds, 40 seconds? Nobody's confused. You got it. You know what he was doing. So this was another slide used in that course. Now, this was a slide designed by the student, actually postdoc, professor, PI, and she borrowed it from him to give her talk on activation of NF-kappa B. Now, the PI, used this slide in a Keystone meeting on NF-kappa B. She tried to use this slide. It took about 5 minutes to get through all of this stuff, but I didn't need to know any of this in order to understand her talk. In fact, I got totally distracted. You know, I was interested in glutamate. So, oh yes, now how does that regulate, what's it do to NF-kappa B? Oh, you know, the other thing is I'm really interested in proteasomes. Oh, I wonder what I-kappa B, I didn't hear a word she said. All she wanted me to hear was that she was studying TNF alpha and IL-1 modulation of signaling and gene regulation. So the only thing she should have put on this slide were the receptors and a dotted line, because there are lots of intermediate steps, and tell me about the genes that were regulated, which was the topic of her talk. So this one's too complicated. The last one was pretty well balanced. So now let's talk about roadmap slides. It's called roadmap slides, outline slides. I actually call them the message slides. They lead the audience through the talk, tells them where they're going. It's the storyline, it's the chapter, it's the verses, it's the headings, the roadmap slides. So here's a roadmap slide I used at a Keystone meeting a number of years ago when apoe was just being discovered as being important in neurobiology. And so they asked me to talk about the history of apoe and the structure and the function related to Alzheimer's, and then some of the early work that we did in cells and transgenic mice. I used this slide several times, just like I used the slide in the demonstration, the talk that I gave you. And each time I came to a new point, I highlighted it in yellow, and then I talked about that. Then I came back to the slide, and then when I presented the in vitro work, I highlighted in vitro studies in cells in culture. So I've shown you now several times how I used my message slide, my roadmap slide, my outline slide in order to lead you through the presentation and the data and the facts. All right, another type of slide are method slides. I've already given you one example of a method slide that makes things be grasped quickly, because you really don't want to spend too much time in an oral presentation on methods, unless, of course, it's really a methods talk. But most of the time you want to get through this as quick as possible, completely for what the audience needs to know, but not too much detail. You want to make it as quick as possible. So here was a slide that was used in that course, in which they were looking at remodeling of dimerystyl phosphatidylcholine vesicles by apoe. And so the presenter, she talked about producing DMPC vesicles, trapping the fluorescent dye in the vesicles, adding apoe, and measuring dye release. And that's good. That's nice, straightforward. But actually, the class said, "Oh, that's a little boring. Can't you do something that's a little bit more interesting to do this?" And so they suggested something like this. It's exactly the same thing. You create the dimerystyl phosphatidylcholine vesicles in which the fluorescent dye is trapped. And when you add apoe, the vesicle reorients and is remodeled, and you measure the release of the dye. Quicker, oh, maybe not, about the same, but more visual. Oh, that's what you're doing. I don't have to think about what do you mean it's trapped. I see it's in the center of the vesicle. Transition slides. As I've already said, when you go from one topic or one set of experiments to another, sometimes even in the background, you need to transition. These are slides that help you change the topic, to introduce a new topic. I use as my transition slides that message slide, that outline, but there are other ways to do it. For example, you can use questions. This was the way a two-part talk was set up in that class, 2 different questions. The actual questions don't matter to you, but the person used them and highlighted them. This was also the transition slide. Now, here's a transition slide that I used in a talk a few years ago, where I described the effect of apoe4 on Alzheimer's disease, but also on traumatic brain injury. And it was a switch to talk about the importance of modifying the structure of apoe4 as a therapeutic target. It was a quick way. I just talked about this. Now I'm going to talk about this. Another question slide, not important to dwell on this. Consider, if you will, the most visual means. We've already considered that, of displaying your method, but now displaying your data. Tables, yes, they can be effective, not usually the best. They tend to get too complicated, but they could be used. Bar graphs, maybe a little better. Figures can be good. Of course, it all just depends upon how clear you make the presentation. For example, here's data again. I select what I have easily available to me. ApoE4 binding to triglyceride-rich particles being blocked by small molecules. And so E4 binds to a greater extent than E3, but if I treat the E4 with the small molecule, now they behave just like ApoE3. That's the point of the slide. It's okay, but this is a better way. More E4 binds to these triglyceride-rich emulsions than E3. But when I treat E4 with these small molecules, they now behave similarly. So just think about ease for the audience. So that's just some examples about presenting data. So let's talk now about some general recommendations for making slides. You all are really good at this. At Gladstone, we actually have a graphics department and some graphics illustrators that can be helpful. But let's talk, first of all, about some general recommendations. Keep it simple. No more than 8 to 10 lines of text, typically 8 words per line. That allows you to have the font size big enough so that you can see it. Use key phrases and use, as I've done here, numbers, or you can use bullets, but set the things off so that they're easily read. Use short phrases to state key points. You don't just read through the slide. I'm reading through the slide because I'm trying to make sure you note everything here, but typically you don't read through a slide. You use color to focus attention on key points, not color for the sake of color, but color for key points, and you use space appropriately. Don't waste a lot of space. Make the font size as big as you can. Remember, the visual is supporting what you are trying to convey. It's not a substitute for your talk. And be sure and proof every slide. Actually ask somebody else to proof. It's amazing how many mistakes you can make. Actually, I think there's a mistake on one of these slides somebody pointed out the last time, and I failed to correct. Maybe you'll see it. Title slides. I think this is really very important. As I've already said, the title's at the top of the slide. It's in larger point size, larger font size. Thus, it provides the initial focus. You always look at typically you look at the top of the slide. So make that title meaningful. And every slide should have a title. Now, capitalization. I think what you're trying to do is make it easily readable, but also you're trying to make your slides consistent. So you want to do the same thing on every slide as far as guidelines go. So I can pick a slide from 10 years ago that I made and put into a talk. You wouldn't know whether I made it yesterday or I made it in 2010. You wouldn't know, because I try to make them all look the same. So titles and capitalization helps you to keep consistent. Now, what I like and what many people believe is the best way for readability is to capitalize the initial letter, only the first letter of nouns and verbs, every word, except don't capitalize articles, a and the, conjunctions, but, or prepositions, all four. Just capitalize the nouns and the verbs. And of course, the first word in a title is always capitalized. And best practices suggest that it's not so good to capitalize all the letters in a title, only, as I show you here, slide titles, capitalize slide, the word, the S of slide, the T of titles. And that helps for readability. Point size, 32 to 36. Make them as big as possible. You want them to be read. Maximum, I like 2 lines at the most. 3 lines is okay, but not too much, because you don't want them to focus on it too long. You want them to get the gist of what the slide's about without you saying anything, and move on. Now, fonts. There's lots of suggestions here. I like Helvetica. It's easy to read. Everything that you've seen is Helvetica. They're all bolded, so that they're easier to read. They're bold, and the font size, titles, 32 to 36. I try to keep the text 20 to 28. I really like 24, 26, 28. Don't make a bunch of different font sizes. It gets a little confusing. And you can use italics or color for emphasis. What you're trying to do is to make a friendly slide that's easy to read. Some people use the Times font, the varying thicknesses of letters, difficult to read. So bold them, they're easier. These are just guidelines, but there's some reasoning behind these guidelines. Color, for emphasis, to draw attention. So when the slide came up, what's the first thing you saw? You saw yellow, number 4, color. Don't use color for the heck of it. Now, I like a dark background. Black, I like. Blue, dark is fine. Other people like white. That's a personal preference. I actually believe the dark background works better depending on the light in the room. It almost always works contrasted with a white text. No matter what the lighting is, it seems to work best for me. That's personal preference, but choose and stick with it. I like black. Use color to focus, I've already said. Use colors that are light, yellow, blue, green, red, and not just a bunch of color for the sake of color. The graphics department at Gladstone did this as a joke for me, because I had just said don't use but 2 or 3, 4 colors at the most, so they put the title of this slide like this. But it does point out a couple of things. One, purple and violet don't show up so loud. So pick from this color palette what looks best. Now here's other examples. I like these light colors because they show up well. Not dark red, not dark blue. I don't like purple, I don't like violet. They're too dark, make things too hard to read. And there are various color palettes that you all know about and that you can choose. So let's talk about formatting of slides. How do you put together a slide for the best readability? I'm not going to spend a lot of time on this. Actually, I'm not even going to go through this. It's here for you to read, but you actually know better than I about how to put slides together. Most of you grew up making slides. I still need help. I know what I like and what I want in a slide, but I'm not the best at making my own slides. But let's look at some examples of what I consider to be good and bad slides. This I consider a bad slide. This was from my postdoc a number of years ago. Now I consider it a bad slide because when you project it in a big auditorium or a conference room, you can't read it. You can't actually even see whether these are pluses or minuses. It's just not a good slide. There's too much black. You didn't fill the space. Just fill the slide. Why waste all of this space here? And so I suggested a slide more like this. Now here, I consider this a good slide. Exactly the same as the last one, but it just is easier to read. There's no question about whether this is a plus or a minus, and that's really important. That you see the difference between a plus and a minus, because a wild type mouse is a plus plus, a knockout mouse is a minus minus. You better be able to see the pluses and the minuses. So I think you will appreciate that the quality of this slide is not as good as the quality of that slide. Now, here was a slide also used in the course. Happens to be, again, about apoe. It's not a terrible slide, but it's just not proportioned right. It's hard to read. It's hard to figure out what goes where. And so we suggested that the slide be made more, well, for one thing, the font size greater, but to use bullets as the first level of indentation and then dashes as the second level. I think you will agree it's a more attractive slide. It's easier to read, and you're pretty clear what is being intended. I think that's correct. Now here's a slide. I also consider this not a particularly good slide. So much wasted space. Fill the slide. It's hard when it was projected in a conference room. We actually couldn't read this ordinate in F kappa B luciferase. They all kind of run together. Couldn't read them. Are those minuses or dashes or what are they? And so we said, oh, come on. Fill the space, make it bigger, make it more attractive. And he actually did. And this is what came out. You can clearly read the X and the Y axis. There's no question about what the symbols are and what the units are. It's very important. So it's really quite important to do this in this sort of way. Now, this is a horrible slide. Was used by a postdoc in a lecture, in a 20-minute lecture. Well, take 20 minutes just to get through this one slide. All he wanted us to see was that he used Cas9 and the signal RNA, and then he looked at the gene regulation under different conditions. So he wanted this, and he wanted this. The slide had all of this. No way to get through or to understand. So only show me what I need to know. Anything you put on a slide should be explained. Don't put something on a slide that you're not going to talk about. He didn't talk about panels B, C, D, E, or F. He talked about A and G. Made no sense. To do that confused everybody. Everybody's looking, oh, what is this GFP stained stuff here? Oh, what is CC, SC? I don't know. What is that? Missed the whole point of the slide. Okay, animations. We've gone through all of these. Now we're to animations. You know about this, you know, stepwise use of points works very well, or an animation of a model. Here's apoe interacting with that same phospholipid, how it remodels the lipid and how it results in a helical formation. This, of course, you all know very well about how to do. And so that's what I wanted to share with you about graphics. I do draw your attention again to the handout that you have been provided. I will draw your attention to the 12 deadly sins, the don'ts of lecturing. If you read those, it will remind you of some of the things that I've said about font size, about what to put on a slide. Crowding too much on a slide is terrible. The use of figures and to illustrate your data. Don't fail to state the message. All of the things that we've talked about, plus questions. And so we have approximately 5 minutes for questions now on any aspect of the lecture that I've shared with you, the comments that I have shared with you. So please do ask questions, and I'll be happy to try to answer them. Okay? Unmute yourself when you ask a question, or if you've put one in chat, we'll take a look at those after we hear from you. But I'd rather start a conversation. Any questions, comments? You don't have to agree with what I've said. That's okay too. I've tried to share with you best practices, but they're your lectures and your skills.
Yoshi has a question, Bob.
Okay, Yoshi.
Hi, Bob. Just one other question. Have you ever had to give a talk where someone in the audience had like a visual impairment, such as color blindness or some other impairment where you had to make adjustments to your talk?
Yeah.
And
yes.
how did you solve that?
Absolutely. Never use red and green on the same slide. I failed to say that when we were talking about the color palette. A significant number of males are colorblind, so that's an impairment that's easily handled, and just needs to be thought about. No red, no green on the same slide. Stick with the light yellow, the light blue, and that tends to work best. And be consistent from slide to slide. For example, apoe 4 is always yellow. That's just the way it is in my lectures. So anytime I talk about apoe 4 and I use color, it's yellow. apoe 3 is blue. And you can mix and match my slides.
Okay, thanks, Bob.
Other questions? I see a hand, but I can't see who's attached to the hand. Please ask.
Hey, Bob. So sometimes there are several slides reach to the same conclusion. So sometimes I will use the title for those slides. I mean, exactly the same title. Is it okay? But it just looks a little boring. But I don't know whether it's okay.
Well, if that's the best way to communicate the information that you're trying to show, yes, I would use it. And actually, there's nothing wrong with being consistent in wording. And I think I said, or I meant to say, that in your message slide in the beginning and the end, use the same words.
Okay.
Don't try to change the wording. It can confuse people like me. I like to hear the same words over and over again. So I don't think that's a problem to use the same title. It's probably better than mixing up the words and making me wonder if something changed. I may not have the exact context of your question, however.
Okay, thank you.
Mm-hmm.
Neil has a question, Bob.
Yes.
Hey, Bob. Great talk.
Neil.
I've seen some people do this a couple different ways, but I was just curious, like, what your opinion was about maybe the best way of highlighting work of collaborators that's like maybe scattered throughout the talk.
Yeah. More and more, and I like it very much. and actually I've started doing it in my talks. I didn't give you an example of this, but I've actually used a small picture and the name of the individual under when I had presented a certain piece of work, because I mean, everybody knows I don't do the experiments. Everybody, there's lots of people that do the work, and so I do it that way. I like that. It's sort of a new introduction, and I like it very much, and I've actually started doing it myself. You can do it at the end. You have to know the audience and where you're presenting. Yes, we want to acknowledge everybody that contributed, but sometimes that gets very long and very boring when no one actually knows the individuals. So you have to be sensitive, you have to be understanding, but in fact, you know, going through a list of 25 people at a talk I'm giving at UCLA and nobody knows them, I wonder what the value of that is, other than the photos inserted in the text.
Thanks. Yeah, it's a helpful tip.
Okay, thanks, Neil, for asking. We have 2 more minutes. One more question. Okay. Well, thank you all very much for your attention. I wish we were together in an auditorium so that I could be seeing you and we could have a better dialogue. In ending, I want to point out to you that it's not by chance that we ended with 1 minute to go. And you should always be mindful of the amount of time that the organizer gives you to present, and you should live up to that as a total commitment. I do that, but it requires that I carefully tailor my talks to fit the time slot and what I can cover. So as I said, we have 1 minute left, and I wish you a very fine evening and continued success. It's a great group, and I couldn't be more pleased and more proud to work with Sudha and others to help in the training. So bye-bye, and the very best to you.
Thank you so much for your time today, Dr. Mahley, and thank you all for joining. And we hope that you can join us next week on July 19th for another program with Dr. Mahley about how to give a talk. Thank you all. Have a good afternoon.